Welcome everyone to my channel BioWorld once again. So today with the same chapter, Principle of Inheritance and Variations, that is genetics. I've come with the topic dihybrid cross. So dihybrid, the word itself you understand, di means two, hybrid means two different, so heterogeneous. So cross between two pairs of contrasting character. Two pair, remember, and it falls under Mendelian third law, that is law of independent assortment, which says that when there is a simultaneous inheritance of two pairs of contrasting character, the distribution of factor of each pair in the gamete is independent of the distribution of other pair. So that means they assort separately, they separately segregate. So what Mendel took in this particular cross, he took two pairs. So what was the pair? Let's see, dominating and recessive. We'll go fast, just see and understand. So in dominating was round and yellow, that is color and shape of the seed, whereas in recessive was wrinkled and green. So this was crossed in first generation, that is F1. So how the gamete would be written? So for round, capital R, for yellow, capital Y. For wrinkled, small r, remember don't write W, otherwise the cross would not be contrasting one. And wrinkled, small r, and for green, small y. So it was crossed in F1 generation. So what will be the resulting offspring? This was the gamete, or you can say P1, that is the gamete in first cross. So in Punnett square, let's see what will be produced. Now before that, I missed out. Let me tell you something. We can't write 4 of the gamete together. So we'll take 2. RY, and from here small r and small y. That would be written together in a Punnett square. So let's rub and do it in a Punnett square. How the cross would be? RY crossed with a small ry. Let's see what would be the resulting gamete. In F1, remember we are doing now F1, that is first filial generation. So gamete ry one side, and here small r, small y, small r, small y. So how the cross will go? I think it is understood. Down little, let me do it. Okay, yes, now. So r, small capital Y, small y. Here also the same, from this and this, capital R, capital Y, small y. This and this, capital R, capital Y, small y. This and this, and small y. So this was in F1. So F1, Mendel got all of the gametes, you can see, round yellow. Though it was not homogeneous, it was heterogeneous, but they all looked like round yellow in first generation, as we found out in which cross? Monohybrid cross. In first generation, all the dominating trait has come out, and recessive failed to express. So phenotype was 1:1:1:1, that is 4 type. Even genotype 1:1:1:1. So all were round yellow plants. So now Mendel has to bring out his separated character, that is law of independent assortment says that only. So he took out possible gamete out of this production of the sibling. That means the produced sibling, the combination of gamete and then their cross will result ultimately the resulting sibling. Let's see how. So let's take out the possible gamete. So what will be possible gametes? So let the combination starts with capital R, capital Y, then capital R, small y, then small r, capital Y, and then small r, small y. So this was round yellow, this was round green, this was wrinkled yellow, and this was wrinkled green. So this was possible gamete formation that we are going to make 16 square boxes Punnett square and cross it to find out the resulting offspring. So this were the gametes, that is possible gamete formation out of the F1 resulting product. So let's see, we are going to do now 16 square Punnett square. I have to make 16 square, little bit short or small, you have to see it. For gamete, here one side, then 1, 2, 3, then 1, 2, 3, and 4. Let's write the possible gametes here. What were the possible gamete? In both sides you have to mention it. That is R, Y, R, small y, small r, capital Y, small r, small y. In this way, you have to fill up the Punnett square and then write the combination. So have you seen how I have written the gametes? So in this way, the lower one also you have to see how I have written so that you can make the boxes properly. Just see, okay? Then I am going to write the gamete by combining them. Let's see. So from both the sides you have to fill it. So capital RY, capital RY. So in this way you have to write capital R, capital R, means capital should be first. Then here this two, capital R, capital Y, small y. Then this two, capital R, small r, capital Y, small y. Then this last one, capital R, capital R, capital Y, small y. So in this way we have to fill all of them without doing mistake so that the gamete formation turns correct one. You have to match the combination. See how I am writing it? So have you seen all the combination I have written there? So in this way both the side gametes have to be filled. So now phenotype, if you write down properly under it, that is under the genotype, it will be easier to calculate. So phenotype means, see, wherever the capital is there, remember it is dominating. So this one will be round yellow. just go on writing in a small short form, round yellow. This is also, you can see, whenever there is a capital, you have to understand that it will be dominating character, expressive one. Here also round yellow. Here also round yellow. So you can see that dominating is coming many in compared to the here. Now here you can see little change, 2 capital R's, 2 small y. So that means one will be round, another will be, Y was for green. So remember, this one is round green. This is again round yellow. This is round green. This is round yellow. This is also round yellow. This is smaller. Remember, no capital R, so it will be wrinkled yellow. This is also wrinkled yellow. In this way, you have to understand the phenotype. Downward also round yellow. This is round green. This is wrinkled yellow. This is wrinkled green. So I think you have understood how the character got assorted independently here. That means wrinkled green has also come out. That is, recessive pair also has come out. Round yellow, the dominating has come out. Then round green has come out. And wrinkled yellow also, the character got assorted independently in compared to the other one. So let's count how many was round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. So I'm writing here RY, that is round yellow, was 9. Now next was round green. How many was round green? See, one here round green, one another here and there. So 1, 2 and 3. Then wrinkled yellow, how many? Just count and see. Wrinkled yellow, one here, 1, 2 and 3. And the last pair, that is the recessive, was only 1. So you can understand the ratio has come up to 9:3:3:1. This is the phenotypic ratio. And if you go to genotypic ratio, it will be big one. Individually, you have to segregate. So what was the phenotypic ratio in dihybrid cross of Mendel? It was 9:3:3:1. That is 9 round yellow, 3 round green, 3 wrinkled yellow and 1 wrinkled green. So in this way, the character got segregated, or the factor got distributed independently in the second filial generation. So this was F2, and if you are asked in the exam, write down the phenotypic ratio of F2 generation. It will come up to 9:3:3:1. So this was dihybrid cross of the Mendel with the law of independent assortment. Hopefully video would be beneficial. Thank you for watching it. Do like, share and subscribe my channel. Thank you.